A warm greetings to all my viewers from Dr. Raktim Mukherjee, Assistant Professor and Head of the Department, Shri P.M. Patel Institute of PG Studies and Research in Science affiliated to Sardar Patel University, Anand. In the previous video, I just introduced you to signal transduction pathway, of which our first video referred to GPCR, that is G protein coupled receptor signaling pathway, and our first example in the earlier video was epinephrine signaling pathway. Epinephrine is a very important life-saving hormone, and we also studied that epinephrine increases the heart rate. Now, very interesting question arises: what would happen if this epinephrine signaling pathway is kept on and on and on, and if that happens, it would have catastrophic effect on our cell? So it doesn't happen. What happens is that the epinephrine signaling pathway is switched off. So the title of my today's talk is how epinephrine signaling pathway is switched off, and this is the visual background which will serve as a template for today's talk. Just as we switch off any electrical circuit by powering off the switch, in similar terms, our question is how this epinephrine signaling pathway is switched off. That is what we are going to study in this today's very important pathway. Before we begin, we once again get you back to epinephrine signaling pathway. You have epinephrine binding to beta adrenergic receptors. So on binding to the ligand, the receptor activates G protein, and once the G protein is activated, epinephrine binds to the beta adrenergic receptors. It activates the G protein, and this G protein in turn activates the membrane bound enzyme, the adenylate cyclase, and this is done by exchange of the GTP for GDP. So adenylate cyclase, once activated, this adenylate cyclase acts as a second messenger, the cyclic AMP. This increase in cyclic AMP result in a biochemical response to the initial signal, and this increased cyclic AMP leads to conversion of inactive protein kinase A to active protein kinase A. Now, there is a very important question which we are going to ask to this pathway, and the question is: first, we have an external stimulus. This external stimulus leads to epinephrine signaling pathway, and this epinephrine signaling pathway creates a physiological response. But after the signaling gets complete, that is post-signal transduction, the question is, how does the cell shut down the epinephrine signaling pathway? That is the major question that we are going to address in this video. To this end, let's visualize the diagram. The diagram that there is an external stimulus. This external stimulus leads to, you have GTP attached to G alpha, and as you can see, the GTP is attached to G alpha protein. The GTP is attached to G alpha protein. This is where after the exchange takes place, the GTP is attached to the G alpha protein. This is your alpha segment, and this G alpha protein will remain attached to the adenylate cyclase. This portion of the diagram, you can see GTP bound with the purple colored alpha fragment is bound to the membrane bound adenylate cyclase. This way, once GTP bound alpha form is bound to the adenylate cyclase, the adenylate cyclase will continue to produce cyclic AMP molecules. And you know, cyclic AMP is the one which converts inactive protein kinase A to active protein kinase A. So this was the story from the epinephrine signaling pathway. But later what happens? Later GDP attaches to G alpha protein. Once GDP is attached to the G alpha protein, that is in the initial part of the pathway, the G alpha protein will be detached from adenylate cyclase. So this is very important. Whenever GDP is bound to alpha, it remains detached. I will show outward arrow, that is detached. But whenever GTP is bound, it remains attached. I show inward arrow, that is attached. So one important clue we are getting from this figure, that if we have to remove or if we have to stop the epinephrine signaling pathway, we should not allow GTP to bound to G alpha. Rather, we should allow GDP to bind. Once GDP binds, automatically this heterotrimeric complex will detach from the membrane. And for this purpose, we have to understand the role of intrinsic GTPase activity. What I mean by intrinsic GTPase activity, you have an intrinsic GTPase activity of G alpha. This G alpha has an intrinsic GTPase activity. So G alpha protein has the ability to hydrolyze GTP into GDP. Once GTP is hydrolyzed to GDP, this is called as the intrinsic GTPase activity of G alpha. Why it is called as intrinsic GTPase activity? Because this GTPase activity occurs within the cell, and so it is called as intrinsic GTPase activity. Try to understand, this intrinsic GTPase activity occurs within a few seconds or few minutes after the G alpha protein is activated. Now you might be asking yourself a question, why it doesn't happen immediately? Why it is giving few seconds to few minutes time? The answer lies in the next point, because it gives time from few seconds to few minutes, that is to give plenty of time, sufficient time for the cell to finish or carry out the epinephrine signaling pathway. So our idea is, let the cell first complete the epinephrine signaling pathway. After the epinephrine signaling pathway is complete, the cell should not remain active. It should immediately switch off the epinephrine signaling pathway, and one of the important way that it does is by intrinsic GTPase activity. Now let's understand how the dissociation of epinephrine takes place. Now how can the cell inactivate the epinephrine receptor complex and prevent any further activation of G proteins? So for this we have to understand, when the epinephrine concentration drops, the epinephrine becomes more likely to dissociate. So quite obvious, whenever you have, this is the receptor, this is the receptor, and this is epinephrine molecule. I will label it as E epinephrine molecule. Suppose the epinephrine concentration in the extracellular space drops. Again, since it is dropping, I will depict by downward arrow. So whenever epinephrine concentration drops, this epinephrine, rather than being bound to the receptor, will automatically dissociate, and that is what is being highlighted, that when epinephrine becomes concentration drops, this epinephrine is most likely to get dissociated. And this is important. We want the epinephrine to be dissociated. If the epinephrine becomes dissociated, the pathway would stop, and that was the the question that we started, how epinephrine signaling pathway stops. Now the question is how the signal initiated by epinephrine is switched off. That was the question we are trying to answer, and we already highlighted a very important role of intrinsic GTPase activity. As we see from the snapshot summary, that intrinsic GTPase activity, as the name suggests, GTPase, it will break GTP into GDP and phosphate. So G proteins spontaneously reset themselves to GTP hydrolysis. Intrinsic GTPase activity of G alpha will hydrolyze the GTP to GDP and phosphate. This bound GTP acts like a built-in clock. This is a programmed clock that spontaneously resets the G alpha unit after a short period of time. So what is the resetting? G alpha should ideally be GTP. GTP bound G alpha means adenylate cyclase is activated. After adenylate cyclase is activated, you allow the epinephrine signaling pathway to complete. Immediately after it gets complete, you don't allow GTPase to remain. Rather, you break down GTP by activity of intrinsic GTPase activity. So it's a built-in clock that immediately or spontaneously resets the G alpha subunit after a short period of time. After GTP hydrolysis, the release of phosphate, this is the phosphate, the release of phosphate, the GDP bound form G alpha reassociated with G beta gamma to form the inactive heterotrimeric protein. It means whatever it was earlier, what was earlier before epinephrine bound, it was in GDP bound form. And because of GTPase activity, again it will return back to the GDP bound form. Remember students, GDP bound form will not activate membrane bound adenylate cyclase and it will not form cyclic AMP, whereas the GTP bound form will activate adenylate cyclase and this in turn will activate the protein kinase A, inactive to active. So this inactive protein kinase A is converted to active protein kinase A. So this is the important role of intrinsic GTPase activity. Someone asks you a 2-mark question, what is the role of intrinsic GTPase activity? Intrinsic GTPase activity will help in breaking down GTP into GDP and phosphate. What is the importance? Once GTP is exchanged for GDP, again the adenylate cyclase activation would stop. The heterotrimeric protein will dissociate and that will stop, or it will control the epinephrine signaling pathway. So how the signal initiated by epinephrine is switched off? G proteins spontaneously reset themselves through GTP hydrolysis, so dissociation. And one of the important area of dissociation, that was one is concentration, second is signal termination. So signal termination, as the first, when the hormone dissociates, that is epinephrine dissociates, it will return the receptor back to the unactivated state. So once the hormone dissociates, it will return back to unactivated state, and this unactivated state is favored by hormone receptor complex that activates a kinase that phosphorylates serine and threonine residues in the carboxyl terminal tail of the receptor. Because of the activation of the carboxyl terminal tail in the receptor, you have a diminished activity to activate G protein, and this receptor kinase utilizes ATP and converts to ADP, and the name of the molecule is beta arrestin. The beta arrestin will arrest the action of epinephrine. So that concludes the epinephrine signaling pathway, especially how the epinephrine signaling pathway is switched off.